Efforts to eradicate and treat the terrifying destruction of parasites all over the world meet with varied and intermittent success. In the case of vector-borne parasites, African governments have attempted to eliminate the vectors, the flies and mosquitoes and other insects that carry parasites for river blindness, elephantiasis, and malaria. These efforts have slowed the spread of some infections, but many scientists argue that eradication of parasites may not be possible.
We've actually never been able to control, to the point of extinction, any parasitic infection. The only disease-producing agent we've ever been able to get rid of is smallpox. And that's because smallpox only infected the human and no other host. Some of these parasites can infect multiple hosts. You can have reservoirs out there. So even if you could effect cures in people, you still couldn't get rid of it in the animal populations that might also harbor the same stages. So even if you could control everything in the human population, you could still reintroduce it every now and then because of these animal reservoirs.
If eradication isn't an option, then controlling the spread of infection is the answer to limiting parasitic outbreaks. One control success story is the onchocerciasis control program.
When onchocerciasis was at its height in the late 60s and early 70s, it became evident that this disease was a major impediment to successful development, particularly of these countries in West Africa where it was so highly endemic. Because of that, the World Bank, the WHO, and other international organizations banded together to form the Onchocerciasis Control Program, which has, in its 25 years of existence, had a major impact on blocking the transmission of the disease, protecting the vision of over 600,000 Africans in West Africa, and allowing somewhere in the order of 15 million children in this area to be able to grow up without the threat of blinding onchocerciasis.
The guinea worm story is one of our shining lights in terms of parasite controls. It's found throughout the tropics, throughout Africa, throughout the Middle East, throughout India, and even in South America as well. It's an intermediate host, that is, it lives in other things besides humans. It lives in this little water flea. And by filtering out the water flea, you can get rid of the guinea worm. And we taught people how to do that. They went and did it. And we've controlled this infection now in all but 13 countries throughout the world. It used to be in hundreds of countries.
In the developed world, new water treatment practices have been developed to keep out Cryptosporidium parasites. Milwaukee spent $80 million building a new water plant. But the tragedy in Milwaukee brought attention to the daily efforts being made on the part of water treatment officials to ensure that our drinking water remains safe.
The lessons that we learned in Milwaukee is you need to be prepared. You need to know the capabilities and the vulnerabilities of your water treatment system. One can never say this will happen again, but one can never say it won't. And so until there's assurance between the health department, the water officials, and others that you're as prepared as you can be, you're at risk.
David Leiby of the American Red Cross believes the Milwaukee outbreak was a wake-up call.
The sheer size of the incident suggests that there's a potential for this type of thing to occur in the future. I mean, even though our water supply, like our blood supply, is very safe as well, and great measures are taken to prevent these kind of instances or these kind of occurrences from happening. However, it just points out the fact that parasites can, in fact, be very opportunistic. And then if there is a possibility of them getting into a system, in this case a water system, which they are allowed an opportunity to grow and develop and to replicate, then they certainly can take advantage of it.
Some see a future in biological controls, putting parasites at war with each other. Introduce a benign echinostome fluke to the host of a parasite like the schistosome fluke, in hopes that the benign fluke will attack and kill its harmful cousin.
The snail is infected with this evil schistosome, and along comes this nice echinostome that has a mouth and a gut, and it seeks out the schistosome in the snail, and whammo, it sucks it up, it swallows it, and the schistosome larval stage is gone. This is a marvelous biological adaptation whereby this tug of war is going on in the snail, and the schistosome, this evil schistosome, is destroyed by this nice kinestome.
The idea of biological control in some respects is very attractive, as opposed to something like DDT, which is very harmful to the environment. If you could have a biological control, for instance, another animal eating the intermediate host or one of the vectors of a parasite, you know, that's a great idea, because that doesn't involve something that generally harms the environment or disrupts the ecology, at least in most cases.
Another avenue of protection is the development of a vaccine against parasitic infections. Dr. Stephen Hoffman and his team at the Navy Medical Research Center in Bethesda, Maryland, are developing groundbreaking vaccines for malaria. The team begins with thousands of Anopheles mosquitoes from the research center's insectary. Volunteers are bitten by malaria-infected mosquitoes that have been exposed to radiation. The radiation weakens the malaria parasites harbored within the infected mosquitoes. The weakened parasites evoke responses from the volunteers' autoimmune system. After thousands of these responses, the volunteers develop immunity to malaria. While progress towards a workable vaccine is made every day, the complex nature of parasites creates a moving target for medical science.
They have multiple antigens or proteins that are expressed on their surface at different courses in their life cycle that makes the design of a vaccine almost impossible.
The more we learn about these parasites, the more respect we gain for their side of the life cycle, and the more respect we gain for the immense amount of selection those parasites have caused our biology to undergo.
The ability of these organisms to infect new hosts and infect virtually every animal that we know. There's just so many different varieties of parasites and so many complex things that you can learn, as well as simple things.
There's nothing easy about studying parasitic organisms. They tend to be very difficult to maintain in the laboratory. It tends to be very difficult to get large numbers of organisms to study. As a result, doing biochemistry on these organisms is difficult. Doing genetics on these organisms are difficult. Parasitologists, scientists who study parasites, tend to be of a very special breed. There's something else that motivates parasitologists to study these organisms that would not ordinarily motivate a person studying another class of organisms.
The wonder of it all and the complexity of what we see in these organisms, going from single-cell organisms to very complex organisms, from parasites that you have to look at under a microscope to tapeworms found in whales that are 100 feet long.
The ultimate goal of studying parasites and to learn about their biologies is to ultimately exercise some control over the way they spread.
My cousin, Rabbi Filozowski, told me about a Hebrew concept, which he calls tikkun olam, and it means to better the world. It kind of refers to the fact that an individual's obligation in life is to try to want to better the world. And I think that's the one unifying feature I've always seen among my colleagues in parasitology, is no matter what particular parasite they're studying, there is some humanitarian motivation in wanting to study these class of organisms.